Hello student, good morning. To continue the lecture, that is the different types of cells in sponges or Sycon. There are different types of cells found in sponges which are not organized into well defined tissues. The cells form loose coalitions and they act more or less independently of each other. The sponge cells are as follows: pinococytes, porocytes, choanocytes, amoebocytes. Amoebocytes in collencytes, arachnocytes, chromocytes, thymocytes, trophocytes, phagocytes, myocytes, scleroblast, gland cells, germ cells. This is the different types of cells in the sponges. Pinacocytes are thin polygonal scale like cells. They are large and flat with a central nucleus. They lie with their edges touching. They are highly contractile. Pinacocytes. Contraction of expansion of the edges of pinacocytes can slightly decrease or increase the size of the entire sponge. Pinacocytes forms the external dermal layer. They line the incurrent canal and in some they also line the spongocoel. The external pinacocytes are spoken of as ectoderm and those lining the spongocoel as the endoderm. Prosopyle, porocytes. Next, porocytes or pore cells are modified pinocytes. They are large size contractile cells. Through the porocytes runs a large perforation called prosopyle which connects an incurrent canal to a radial canal or to flagellated chambers. Choanocytes, third number. This is the types of cells or sponges. Choanocytes, flagellum and nucleus are present in choanocytes. Or flagile endoderm cells are large oval nucleated cells each with a contractile vacuoles and some food vacuoles. At one end is a long flagellum arising from a basal granules. The basal granules is joined to centrioles and the two together are called as the centroblepharoblast, which controls the movement of the flagellum. Below this is the parabasal body joined by fibrils to the centroblepharoblast and the nucleus. The flagellum has two central fibrils and 9 double peripheral fibrils. It is surrounded at its base a transparent contractile collar made of a ring of erect and closely set microvilli which project from the cell. Choanocytes forms a layer of adjacent cells in radial canal or flagellated chambers or sponges. The movements of their flagella causes a current of water. Choanocytes are endodermal in origin. amoebocytes. They are amoeboid cells with pseudopodia. They wander about freely in the mesoglea. They are modified to form the following types of cells. Coleocytes have several slender, long branching pseudopodia. The branches are like connective tissue cells in the mesoglea. Archaeocytes are large amoebocytes. They have a few blunt pseudopodia. The nucleus is large. They are generalized cells and they transport food and waste substances. They can give rise to other types of amoebocytes. They form sperms, eggs, and asexual reproductive bodies called gymules. Such cells, which can change into almost any kind of cells within animals, are said to be totipotent. Chromocytes are pigmented amoebocytes having lobose pseudopodia. They probably import coloration to the body of sponge. Pigmentation color. Thysocytes are amoebocytes having lobose pseudopodia like chromocytes but filled with the food recenses. Thus, these cells work as storage cells, food vacuoles like food particles. Thysocytes. Trophocytes are found loaded partly or completed with digested food and serve to transfer it from one place to other. Phagocytes collect food from choanocytes through their pseudopodia and also engulf excreta. Adamantine tissues. Myocytes are fusiform contractile cells. They form a sphincter around apertures such as oscula and apophyses, where they are like muscles to open or close these apertures. They show some similarity to involuntary muscles in shape and contractility. Myocytes. Scleroblasts are amoebocytes in the act of secreting the skeleton of sponge. They are called calcoblasts if they secrete calcareous spicules, or silicoblast if they secrete siliceous spicules, or spongioblast if they secrete spongy fibers. Triradiate spicules. Scleroblasts. Gland cells are amoeboids with long strands at one end. They are found attached by their strands to the surface of sponge. They secrete slime. Germ cells are male and female gametes, sperms, and ova, different from archaeocyte cells. In some cases, the germ cells are said to be differentiated from choanocytes. Tail head sperms ovum. This is the different types of cells in sponges or Sycons. Different types of cells. pinocytes, porocytes, choanocytes, amoebocytes, collencytes, archaeocytes, chromocytes, thiazocytes, trophocytes, phagocytes, myocytes, scleroblast, gland cells, germ cells. All these are the 10 types of cells are present in different types of cells in the sponges. Okay, thank you.